pleasure to introduce Dr. Inge Holtman. Inge, we know each other, I think, for more than 9 years, and it still feels more like yesterday, not really like 9 years, which is a good sign. So Inge is now an assistant professor and group leader, or Rosalind Franklin group leader in Groningen, University Medical Center in the Netherlands. And this is also the place where Inge started her career in science, or at least her PhD career. She joined the lab of Dr. Bart Eggen, focusing on microglia, the most interesting cell type of the brain. We all agree on that. But early on, she already focused or saw the potential of genomics. So the computational analysis and where this could bring us, how this could potentially make us understand how certain genetic predispositions can lead to neurodegenerative diseases and also new psychiatric diseases. So already during her PhD, she was focusing on computational analysis and was looking at the gene expression signature of microglia in the context of aging, but also of Alzheimer's disease, ALS, Parkinson's disease. And first she was working on mouse, and already there she was very successful, had a great impactful paper. And also back then, and this feels like old times, but already back then she introduced a database. So people like me who were not very proficient in bioinformatics or computational analysis would actually use the browser to look at gene expression. So that was instrumental very much for me back then. That was mouse. She then moved on to work on the human species, a little bit closer to us, so to say, and also was working on microglia, also was looking at gene expression, analysis of microglia isolated from the cortex of human individuals. And pretty much at the same time as Chris Glass published his work on human microglia and epigenetic signatures, she also put out that work in Nature Neuroscience. And gene expression, we know, we love it, but we also want to understand how do microglia think, what makes a microglia, what is the epigenetic regulation of microglia. And there's only one place in the world where you can really study this, in my opinion, or the perfect place actually in the lab of Chris, who really merges omics and macrophage biology. And so she decided to pursue her postdoc in Chris's lab, and that's where we met 9 years ago. And her successful career path really went on by looking not only at microglia, but extending the work to astrocytes, neurons, and oligodendrocytes, looking at what are the transcription factors, what are the active gene regulatory elements that drive those cell types in the human brain. And also in that paper, very instrumental at that time was really to assign certain genetic risk variants to a cell type. So in the form of microglia, a lot of those risk SNPs for Alzheimer's disease, they're actually located in active gene regulatory elements of microglia, not in neurons, not in astrocytes, but mainly in microglia. And the second major advancement that Inge made was really to analyze plug-seq data or chip-seq data, which really can align or assign the risk variant to the respective gene. So back then, we just looked at the nearest gene to the risk variants, but Inge was actually able to assign risk variants to the particular gene. So these were two major advancements, and that was published then together with Alexei Knott in Science in 2019. So that was a huge success. And unfortunately, when you have a lot of success, you become very popular, and especially Groningen, where Inge comes from, where she did her PhD, they were calling back and were asking, when do you come back? And back then in 2020, Inge decided to go back to Groningen, which was a very unfortunate and a big loss for us here at UCSD. But yeah, man, the Groningen people, they got lucky. They got Inge with all her expertise and also her expertise in then machine learning and AI that she really then proceeded to pursue there in Groningen. So she founded the Netherlands Neurogenomic Database in collaboration with the Netherlands Brain Bank, which is a very successful brain bank with lots of thousands of brains, also with a lot of clinical data. So they have always beautiful clinical presentations along going with this, but also with neuropathology and also with multi-omics. And really, her expertise there was to integrate all this kind of data together, which is very complicated. Usually, we look at one thing and the other, but don't see the whole picture. But really, Inge mastered that. And last year, I think, was super successful. She published 2 papers, one in Nature Medicine, one in Acta Neuropathologica, where she was taking data from the brain bank, clinical data, pathological data, and then using natural language processing to actually define disease trajectories, but also define subtypes of diseases, especially in multiple sclerosis. With all the success stories, there come awards and there's grants. So Inge is successful in both of those things. She got the Rosalind Franklin group leader position and also the fellowship. Rosalind Franklin, if people don't know it, she was actually the person who used X-ray crystallography to determine how DNA looks. We all think of Crichton and Watson, but it's actually Rosalind Franklin who then had the first picture of DNA. Numerous grants. You're very successful. So Inge got a prestigious ERC award in Europe. Inge is a work group leader in the Institute of Chemical Neurosciences, which looks at the what are the underlying mechanisms of neurodegenerative and neuropsychiatric diseases. And recently she was awarded the Jan Zuckerberg Initiative Award. And Inge just returned from San Jose, or was it San Francisco? San Jose, where she was at the initial meeting where they talk about how you can incorporate AI with brain bank data. And this, I assume, is also what Inge will talk about today. So I'm very thrilled to have you here. With all those accomplishments, I also would like to recognize that you also have other accomplishments. I always remember the last Christmas party at Chris's house, where I think you did an impromptu piano concert, which was, I still remember it very fondly. We had just another Christmas party a few months ago, and the piano was alone. There was no Inge. So yeah, I'm very thrilled that you're here. I'm very looking forward to your presentation. Thank you.
Well, thank you, Johannes, for a very kind introduction. I think that's yeah, one of the most extensive introductions I had in my career, so that was already applaudable. Yeah, so it's great to be back in San Diego, my postdoc lab, and San Diego still feels like home. I'm walking around here and I feel like, you know, Californians, I think, are the friendliest people in the world, very respectful people, and it's just such a pleasure to be in this place and discuss science with so many excellent scientists. So also really thanks to all of you for being here today. See? Yeah, so before I talk a bit about my work, I also like to zoom out a little bit. And I think in general, we know that if you have a random group of people, such as ourselves, many of us will develop a brain disorder. And unfortunately, it is incredibly hard to predict who will get which disorder. And if somebody gets a brain disorder, it is incredibly hard to determine how the disease will progress. With all of these diseases, there's tremendous variability. Some people go very fast, some people go very slow, and we don't really know what those factors are. And there's still a lot of work to be done to better understand this. One of the major difficulties is that there's extensive clinical heterogeneity. So patients with the exact same diagnosis can manifest very different neuropsychiatric symptoms. When the symptoms happen, the trajectory of the symptoms is also highly variable, and there's still a lot there that we don't really understand. In the same way, if you look at the neuropathology, so each of these disorders is characterized by different neuropathological features, but also there's tremendous variability. There's sort of generic disease processes, but a lot of people have disease processes in other brain regions, other kinds of factors contributing. And so yeah, the variability is really quite a big issue. And it also leads to the fact that the relationship between the clinical manifestation of a disease and the biology is often not clear. I will also show that in my presentation later today, but frequently when a neurologist comes to the conclusion that an individual has Alzheimer's disease or Parkinson's disease, and later this person dies and you perform a neuropathological examination, you will come to the conclusion that they're wrong approximately 20% or 30% of the time. Because of this issue of the clinical manifestation and the biology not being aligned. And in order to learn more about this heterogeneity at these different levels, I think the Netherlands Brain Bank has been a unique institute. It was founded back in 1985 by Dick Swaab. And since that time, we've been able to perform more than 5,000 human brain autopsies. And what is really cool about the Netherlands Brain Bank is the Netherlands is a small country and we have an excellent infrastructure. So we have an incredibly short post-mortem delay. on average only 6.5 hours between the moment of death and the moment that the autopsy starts. So also that leads to really high quality material, and this material goes everywhere throughout the world. It's been sent from Japan to Brazil to the US, and many researchers have been making use of this resource. And with patients with any kind of diagnosis that you can think of, you have a large cohort of individuals, Alzheimer's disease, frontal temporal dementia, progressive supranuclear palsies, many people that have sort of a dementia, but it's not really clear what they actually have. So they have a sort of a comorbid mixed form. You have psychiatric individuals and also a lot of other disorders as well. And the Netherlands brain bank has been very valuable for other people, but it never really been studied in its totality. And that was the aim that I did of a project that I developed when I was a postdoc here, is that we thought like, okay, maybe it's time to start mining this resource in its completeness. So there's a project I developed with the director of the Netherlands Brain Bank, Inge, as well as another prominent PIs in the Netherlands. So we've taken these brain tissues, these data from these brain tissues, with these individuals that I discussed, and we've been mining different kinds of data. So on one hand, we're looking at the clinical data, and as Johannes explained, we have a lot of clinical information. We go back to the medical practitioners, we go back to the hospitals after a person is deceased, and we retrieve all of the medical files, which sometimes literally goes back 30 or 40 years before death. So for very long periods of time. And in the same way, they also do very extensive neuropathological examination. On average, we look at 15 different brain regions with a number of different immunos. And all of this is then summarized in these medical files. And the idea is that let's start using large language models to get this data out and start integrating it with genetic risk as well as with multi-omics data types. So if you think about the clinical history and the clinical information that we had, there was indeed work published last year in Nature Medicine. We developed this pipeline. So on stage one, as you can see, are these medical summaries. So at the NBB, the examination is before neuropathological examination. The healthcare records were being summarized, so everything is translated in English. And you have these summaries that describe on a year-per-year basis what the medical history of that patient was. And that was summarized into these NBB donor files that are being sent to everybody who's making use of this material. And back in 2020, during COVID times, I just returned to the Netherlands. I was locked in my house. You know, I had this great time in San Diego, and now I couldn't go anywhere, as all of us. I was like going through these medical files, thinking, what is actually in here? And I start identifying different neuropsychiatric signs and symptoms, and I start playing around with like large language models or natural language processing methods to like get the model to score whether a particular sign or symptom was manifested, yes or no. And that worked actually pretty well, and it was fun. And then I got like bioinformaticians, like a bachelor program, master program, and I got a technician, and we all started working on this. And at the site of the NBB, we started labeling this data. And it became bigger and bigger, like how many features can we actually identify? So first you have a few, and then you go more in depth, and you say, like, well, we want to go cross disorder. It needs to capture every disorder, brain disorder that we can think of. We also need to have the right level of resolution. So it took us a lot of time to ultimately start building an ontology. Let's formalize the structure of what we learned, labeled a large number of sentences within these individual signs, positive observations, negative observations, and then train large language models and natural language processing AI models to identify whether those signs and symptoms were manifest in a given sign or in a given sentence, yes or no. And together, that resulted in what we refer to as clinical disease trajectories. So it's a Boolean matrix, and on a year-per-year basis, you can say what kind of neuropsychiatric sign and symptom was observed, yes or no. So that if you have that over the whole lifetime or for over really long periods of time, it's really giving very in-depth information on the phenotypical background of each specific donor. And once we had those data and it started working, we obviously came to the point like, oh, what can we now do with it? And we have a number of use cases, and I'll go more in depth on a little bit on, like, we looked at the diagnosis accuracy. We started to see, could we predict the neuropathological diagnosis, use these trajectories? Now, can we maybe also identify clinical subtypes? So to get to the first point of the defining of the signs and symptoms and labels of sentences, it's really a tremendous amount of work that went into it. There are existing ontology structures, but none of the ontology structures was really used for this specific use case of cross-brain disorder research. So ultimately, here you can see all this neuropsychiatric signs that we identified in different domains: psychiatric, cognitive, motor, general. And we grouped them, and you can see different attributes like agitation, restlessness, change behavior, psychosis, paranoid, visual impairments, ataxia, loss of coordination, and on and on. And for each of them, we ultimately also defined what exactly do we refer to, do we mean by this? Because sometimes there are little nuances in what is actually a depressed mood. So we fully, based also on literature, build data to support this. So here you can see a description of what, in our case, depressed mood specifically entails. We cross-linked it to other ontologies, and we gave inclusion and exclusion criteria. And all of this information is accessible on our website, the nnd.academy. So ultimately, we build a lot of ontologies because, yeah, in this realm, there really wasn't much to support. So build an ontology for the clinical history, so these neuropsychiatric signs and symptoms. Also neuropathological diagnosis, right? There's the ICD-10 code that's beautiful for clinical diagnosis, but there really isn't an equivalent for neurodegenerative neuropathological diagnosis. And also in a formal structure. So we built an ontology there, and we also modified the human disease ontology in a way that it could also incorporate its better resolution at the level of neurodegenerative disorders. And then we started building these natural language pipelines. So here you see a bit of the workflow. And the first stage is the model optimization. We took these labeled sentences, performed cross-fault validation, cut the data set in a training, test, and validation set. I'll optimize the whole number of models like Bag of Words and PubMed Birds and T5. These are all state-of-the-art at the time. Obviously now with the GPT-based models, well, many more new models have come out in this field. It's going incredibly fast. But ultimately, we retrained the best model on the full corpus and allowed that to predict on our full corpus what neuropsychiatric signs and symptoms will manifest, resulting in these clinical disease trajectories I was alluding to earlier. Yeah, the Boolean clinical disease trajectories. And then sort of, well, we had done this tremendous amount of work. We were proud of ourselves, and we submitted it to Nature Neuroscience, thinking like, oh, it's so cool. And then one of the reviewers was, yeah, it's very cool. But, what do we actually do with this? Like, what is the neurobiological implications? And we were a bit like, yeah, okay, we hadn't really thought super far in that direction. We thought it's very important for brain research, neuropathological brain research, to have this kind of information. But now, what did we learn in this specific project? Well, I also like to slow this slide because it kind of exemplifies the kind of information that we have. Right, so this is one donor, and this person died in a particular year, and here you can trace back like what signs and symptoms were identified by the model. Right, so here was depressed mood, here was psychosis, here was agitation, suicidal behaviors. Well, you see basically these sort of phases in which this person was manifesting a lot of symptoms. And what kind of symptoms were being manifest. And we're still kind of working on like, what can we do with this? But the first thing that we did is just flatten the vector. Let's see, we take the time component out, and we just calculate the number of signs and symptoms that were manifest in each donor, and then conglomerate that at the level of the diagnosis. That's the first pass. So if you look at control, for example, so here we have the y-axis, we have different diagnosis like schizophrenia, bipolar disorder, major depressive disorder, Alzheimer's disease, Parkinson's disease, dementia with Lewy body, frontal temporal dementia. So these are the largest groups that we have. And then you can see what kind of signs and symptoms manifest. The size of the dots correspond to the proportion of individuals in whom we identify the symptom. And then the intensity corresponds to the number of observations, the mean number of observations we had among the donors in that specific group. And what you could see here, for example, is that bipolar disorder is very clearly characterized by mania. Pretty much everybody who was diagnosed with bipolar disorder exhibits the sign of mania. Well, obviously, that's what you would expect, right? That is what defines bipolar disorder. But it's still nice that we found it back. And in the same way, also depressed mood, for example, is really high in bipolar disorder individuals, and also very commonly identified in almost all major depressive disorder cases. But the nice thing is that also you can see that considerable fractions of cases with all these other diagnoses are also manifesting depressed mood. And I think that's I've heard a lot of clinicians ask this question, like, why do a lot of people with multiple sclerosis, Alzheimer's disease, as well as many other disorders develop depressed mood? Is that something genetic? Is that something about their neurobiology? And those kinds of study questions we could really not ask ourselves in the field of postmortem research before we had this kind of data available. And I think that really exemplifies why this is so important. And we see all kinds of other symptoms, right? So you see compulsive behavior, agitation, and this is only the psychiatric symptoms. And we have many more. And we're literally looking at these lists for hours. There's so many interesting profiles here where we see all these extrapyramidal signs like tremor, rigidity in the Parkinson's disease patients with dementia, as well as progressive supranuclear palsy, as well as multiple systems atrophy. And in multiple sclerosis, for example, we really see like mobility problems, muscular weakness. And in the end, we also performed an overrepresentation analysis for the kind of symptoms you would expect versus the kind of symptoms that were identified. We found a massive overrepresentation, also further corroborating the validity of the approach. And they gave us a wealth of clinical data. We also started looking at the temporal components. What can we actually do here? And for example, here is a symptom, bradykinesia, slowness of movement. It's very common in many Parkinson's-like disorders. And we can plot what is the distribution of when this particular symptom was manifest. And we see, for example, in cases with multiple systems atrophy, is also among the alpha-synuclein family, that this particular symptom is at a much earlier age manifest than in any of these other four disorders like PSP or PD or P30. And we can also then, for example, perform a survival analysis. So what if you manifest a particular symptom? How long do people on average in your category still have, let's say, maybe that's a bit of a crude way of phrasing it, but the interesting thing there is that you could see that the pattern is very different. So when we see that the MSA cases are relatively young and the other cases have a much later age at onset, the distribution of the survival shows that after the symptom is manifest in PSP, DLB, and MSA, the years of survival is much shorter than in Parkinson's disease. And I think it also corroborates the fact that there's these questions on, even though they have all our alpha-synucleopathies, could there be that there are different neurobiological processes among these different alpha-synucleopathies? And can we use the phenotypical data sort of as a readout of better understanding these kinds of neurobiological processes? That's obviously just a hypothesis, but these are the kind of questions that we can really start asking ourselves now. And also in this cohort, we cleaned up all of the clinical diagnosis. So we now have a tremendous amount of clinical diagnosis run through these ontologies, linked to the ontologies, instead of handwritten notes, what they were before. And that also allows us to do all kinds of analysis. Here, for example, we were just looking at like let's look at what the clinicians think somebody had and what the neuropathologist thought somebody had. And we can plot that, for example, for Alzheimer's disease. And then we can see that out of the 625 cases, these 625 individuals, 401, the neuropathologist and the clinicians were in agreement. But you also see that the Jaccard score, or the Jaccard score of 0.642 is actually quite low. There's a whole number of individuals where the clinicians were pretty sure that this was Alzheimer's disease, and it turned out not to be Alzheimer's disease. And similarly, there were also quite a few cases where the neuropathologist neuropathologically defined something as Alzheimer's disease, but the clinicians missed it. And this is not unique for Alzheimer's disease. We see the same profile for Parkinson's disease, as well as for many of these other neurodegenerative disorders. And that's interesting, right? That begs the question, like, why is it so hard to clinically diagnose a neurodegenerative disorder? Like, what makes it so hard? So we thought, well, now we have massive amounts of data, and maybe we can like build our own models, and maybe we can do better than the clinicians, right? That was a little bit of the game, and obviously there's a little bit of arrogance there, I guess. But sometimes in science you have to just try to do something. So we took these individuals that are very clearly defined cases, 2,150 donors, again, run it through a coarse fault validation, use the model, a gated recurrent unit, to optimize optimization, again, a test cohort, got good F1 scores, optimized all of this. And this is a model that's really developed for time series data with missing values. And here you can see the results. So on one hand, we see the neuropathological diagnosis, and on the other hand, we see what the group D predictions are. And if the diagonal is very, bright color, then, it's highly consistent. And if it's off of the diagonal, it's not so consistent. And you can see a number of things, right? So first of all, control cases are relatively easy to identify, but even there are some mistakes. Alzheimer's disease, definitely a lot of mistakes. And also sometimes they're missed, for example, with individuals that actually have a double diagnosis. That makes sense, right? That's definitely very hard. So it's not so strange that the neuropathologist or that the clinicians missed it. But also sometimes it's just another dementia, like vascular dementia, frontotemporal dementia, or sometimes even like progressive supranuclear palsy or some other disorder like that. And for some of the disorders, it really didn't work at all, like vascular dementia. And it seems to be that there's quite a correlation between the number of individuals that we have. So the larger the cohorts, the better the model is able to learn the underlying features. But there also seems to be a case that it is sometimes really quite hard. And if we start comparing now all three diagnostic labels, so the ones from the model, the ones from the clinicians, and those from the neuropathologist, we found out that, well, the clinicians and the model are often, these 95 individuals, both of the model thinks that this is Alzheimer's disease, as well as the clinician thought it was Alzheimer's disease, but the neuropathologist says it's not Alzheimer's disease. And that's interesting, right? So it seems to suggest that there's a subset of individuals that sort of manifest the clinical profile that's really associated with the disease, but then actually they have a different kind of disease. And so we started looking at it in more detail, and we think that we call this that individuals that masquerade as another disorder. And again, you could find that those categories in different disease labels. And that brought us to the question, like, oh, maybe we're looking at the problem the wrong way. So instead of like looking at what everybody has in common with a particular neuropathological diagnosis, maybe we should just look at it from the clinical perspective, right? We have these clinical disease trajectories. Can we optimize a multidimensional dimensionality reduction approach? Then get the main sources of variation, apply clustering, and then see, can we find data-driven clusters, and how do those relate back to the neuropathological diagnosis? And using this approach, we went through different kind of frameworks. It's always one of those questions, the data is kind of unorthodox. So what kind of approach can we find that actually works well? In the end, we settled for a k-nearest neighbor approach. And here the UMAP just represents the k-nearest neighbors. So we found 6 different clusters, and we gave them these labels based upon like what category was most common, right? So here in pink are mostly like control cases. And then here, for example, are mostly multiple sclerosis individuals. This is mostly the Parkinson's-like individuals, late dementia, early dementia, as well as the psychiatric individuals. So that was already kind of nice. And then we could overlay this with the different diagnostic categories. And as you can see, I think this beautifully sort of illustrates this heterogeneity problem that I started off with at the beginning. As you see cases, for example, in red are frontal temporal dementia cases. And many frontal temporal dementia cases are clearly in the late dementia or in the early dementia group. That's to be expected. But you also see that there's quite a number of cases with frontal temporal dementia in whom the dementia actually strikes really late. So they wind up in the late dementia. And it makes sense that the clinician is then not thinking about frontal temporal dementia anymore. If you have a very typical dementia profile, it manifests at late age and you're demented and memory problems, why would the clinician consider that you have frontal temporal dementia? So we also did statistics on those levels to identify these coherent versus individuals versus the non-coherent individuals to see if we can better stratify those subsets. And it's depicted here as like white stars if there's a general overrepresentation. And indeed, late dementia is primarily characterized by Alzheimer's disease, as well as these mixed other dementias. But we see that the individuals that have frontal temporal dementia, the ones that wind up in that cluster are the ones that are often missed by the clinician. Right, so that's really in line with this hypothesis that if people manifest clinically a different disease than what they neuropathologically have, the neuropathologist and the Group D model are missing it. And I think that's tremendously interesting. And those are also worthwhile investigating in more detail. Like, why is the disease looking clinically as a different phenotype than what the neuropathologists have actually examined? And again, I think that that's the kind of questions that we can now start asking ourselves in the field of neuropathological research that we really couldn't address before. Yeah, so I hope I was able to convince you that clinical disease trajectories of brain donors give novel insights into the heterogeneity of these different disorders and allow, I think, for a whole number of new kinds of research lines that weren't possible before. And then I switch gears a little bit, going towards more the neuropathological part of the story, right? We used neuropathological diagnosis before, but I think, like, why are we stopping at neuropathological diagnosis? And again, there's this tremendous amount of heterogeneity. Maybe there's a better way to sort of quantify each individual person according to the different axes of neuropathology and let go of these neuropathological diagnoses. Right? Who says that maybe also different neuropathologists might give slightly different neuropathological diagnoses, because in the end, these are also only human beings and they all have their internal set of criteria. And those criteria are not always equally consistent among different neuropathologists. So I think that's the other way of looking at the same cohort, at the same data, at the same underlying problem. And this is a paper where we looked at the multiple sclerosis cohort. So multiple sclerosis, a severe neurodegenerative disease. And in this particular situation, they quantified, this is ongoing work from the group of Inge Huizinga, they've been quantifying the different lesions. So MS individuals have all kinds of different lesions, and you can quantify those using different parameters. You can say it's the active lesions or mixed lesions, inactive lesions, remyelinated lesions. You could quantify how many lesions does a given individual have. Are they reactive? Where are they? And we had this massive amount of data on these individuals. And we felt, I had a student in my group, Alice de Boer, and we felt like, okay, maybe we can just apply a factor analysis here and see what the underlying factors might represent. And then maybe we can use other kinds of data as sort of orthogonal validation for further interpretation of what those factors might actually mean. So the factor analysis resulted in 3 different dimensions depicted here. First, we were looking for clusters, but we really couldn't find strong discrete categories. So it really seemed to be more a factor approach than a cluster approach. And we find these 3 factors, and they represent, after interpreting them, we think that they represent really important different parts of the MS disease processes associated with demyelination, immune activity, microglia reactivation, lesion initiation, as well as loss of lesion activity and scar formation. And what that exactly means, you can read the paper in more detail. But the point being, I think that these factor analyses are nice to identify in a data-driven way what the independent disease processes are. But the question is it really always a biological thing, or could you also measure some sort of artifacts? And that's the nice thing, is that we have a lot of other kinds of data. Right? This is clinical data that we just got through the modeling approach I was talking about before. We have genetic information. We also had other neuropathological parameters. We even had a number of immunological parameters. And I'm not going to go into depth that much on the paper, but the nice thing is that for each of these factors that we identified, we found either genetic evidence or clinical evidence or neuropathological evidence really supporting the validity of the approach that we're taking. And I think this is really where I'd like to take the field next, and see if we can do this instead of focusing on one disease, maybe do this on all of the different kinds of dementias and see what the underlying disease processes are across different dementia types. This is still a little bit on the other side. So, for example, dimension 1 of multiple sclerosis individuals also showed a lot of cardiovascular problems. Many of them have autoimmune diseases. Some of them have hypertension. And that's also the nice thing, is that we have these ontology structures that allow us to go through different levels of the analysis. Right? People in the beginning were telling me, oh, you have so much data and you only have so few individuals. How can you ever find any profiles? And this is where ontologies come into play. Obviously, we cannot look at each diagnosis, because how many clinical diagnoses are there? Thousands, right? And we never have sufficient power. But by using the ontology, you can go much higher into the branches. If you start taking higher nodes within those branches, then you don't have this whole multiple testing problem. And you can actually see quite nicely what kind of profiles are relevant. And this is just to exemplify the fact that I think that approach works quite nicely. That we really see independent diseases associated with independent neuropathological processes. In the same way, we look at medication. We also cleaned up all the medication data, again, put in ontology structures, and also medication is there in a lot of different levels of ontologies. And we found that immunomodulatory agents and such were really strongly associated with different clinical parameters, but not so much with our different dementias. But specific categories of medication actually were. Yeah, a little bit on how are we in with regards to time? Okay, I still have some time. Yeah, so this way of thinking about multiple sclerosis and about the heterogeneity across different neuropathological diagnoses, would like to take that further. And for that, I'd like to start off with the histopathology data. I feel like right now, a lot of the analysis that we done in the multiple sclerosis project were done with labeled data by human beings. Humans look at the immunos and they say, well, in this immuno, I see these lesions based upon a number of parameters. But why do we stop there? Why do we not go to the raw data? And the raw data of neuropathologists is ultimately the immunohistochemistry. And there's so many different immunos, right? You have all of these different immunos. You have immunos for amyloid, you have an H&E staining for alpha-synuclein. Some of them are disease-specific, some of them are generic. We do it in many different brain regions. So also there, I think like data-driven delineation of histopathology data is really the future. And that was also why we got funded by Jan Zuckerberg initiative for us to start being part of a network of different histopathological experts, neuropathological experts. Like how can we optimize this process of data-driven way to segment and analyze these kind of messages, images. Yeah, this is a part of a paper that is published by Shivam Kumar. Shivam will join my group as a postdoc next month. And this is work that he did in his PhD. But I think it really quite nicely illustrates that he take this histopathology data, build an encoder decoder model around it, and then combine this data with spatial gene expression data. Because many people do these kind of histopathology data segmentation in different ways, right? Are you interested in amyloid, or interested in tau, or like where are they, right? So that's very much like the human way of looking at it. But I think the other way of looking at it is like use spatial RNA sequencing as an objective readout, because it's known that spatial RNA sequencing profiles are correlating to all kinds of neuropathological approaches. So why do we even need the human being to segment the data to get started off with? And could we not maybe use like spatial RNA sequencing data in the beginning, train AI models to learn which spatial expression profiles are associated with which different histopathological features, predict those back, and then perform like a factor analysis on those predicted gene expression profiles. That's a strategy that we're taking next. As I said, the postdoc is going to start next month. And we're also working quite extensively on genetics, also something I could talk about for a long time. This is all still unpublished, but I think it's nice to show. If you talk about genetics, you have different kinds of genetics. You have like GWAS, rare common variants. You also have rare variants. So we're also doing whole exome sequencing in collaboration with Roel Ophoff here at UCLA. But I won't be talking about that yet because those analyses are still in the early stage. But we did do Illumina SNP chips for also the same cohort for almost 3,000 individuals. And then we have all these variants, and then we can subsequently look at GWAS variants. And it's important to note that most GWASs are done on clinical phenotypes, right? So we know that the GWAS for Alzheimer's disease done is now based on either Alzheimer's, clinically diagnosed Alzheimer's cases, or even by proxy cases. And this is a serious issue because this results in the fact that there's going to be a lot of contamination. We know that a considerable number of these individuals don't actually have Alzheimer's disease neuropathologically defined. And again, you can even ask the question, should we run GWASs on these discrete categories anyways? Can we maybe not move towards a GWAS for a neuropathological trait, which is also work that was published last year in Nature Genetics in a way that I think is like very elegant. We can also use this data, for example, to calculate polygenic risk scores and then see what do those PRS profiles look like in our cohort. This is a little bit of a preliminary analysis, but I think it really quite nicely illustrates also the heterogeneity problem, but then from the genetic angle. So if you took a GWAS data set, so PRS scores for different neurodegenerative diseases, Alzheimer's disease or Parkinson's disease or dementia with Lewy body, frontal temporal dementia, and you subsequently see, how does that relate to our cohort? And here each group is compared to control. we said that Alzheimer's disease GWAS risk is much higher in our Alzheimer's disease individuals. Okay, that's great. I know that's obviously proof of principle that it works. What we also see is that Alzheimer's disease actually significantly enriched in frontotemporal dementia cases, as well as even in cases with multiple sclerosis. And in the same way, we also see that Parkinson's disease is enriched in Parkinson's disease, but also in progressive nucleosuperior palsy, as well as in multiple systems atrophy. So I think that here we have a genetic angle and looking at the same kind of problem, and you again see how the genetic susceptibility of one disease is also influencing, I think, your susceptibility to develop any of these other disorders. And sometimes the original GWASs have not even found genetic correlations. So it's really nice to say that even though the GWASs themselves don't find genetic correlations, we do see these higher order overrepresentations. And yeah, this is just the first pass, and there's much more that I can talk about. Maybe next time that I'm here, I can tell you the full story, but it's going to be much more time than I have for today. But I hope to conclude that I convince you that I think the NND is an important resource to study brain disorders. We need to move from individual data types towards multimodal data types because it's important to identify subtypes of brain diseases the more data you have. By integrating neuropathology and genomics and clinical data, we can much better see the big picture than what we can see if we only look at one kind of data. And that's I think, at this point in time, too often neglected. We have a lot of ongoing activities. We perform detailed genetic analysis. As I said, whole exome sequencing data is coming up. We're integrating multiomics data. We have the large language models that I was talking about. We're still relatively old models. There are newer models. And more importantly, these new models can also work with Dutch. The previous models were all English-based, hence we were restricted to the English summaries. But the summary is just the summary. We literally have hundreds of pages of medical information for each donor that is still in Dutch. And with like ChatGPT and MedGemini and other kind of large language GPT-based models, you can now go towards the original medical files and generate what I think will be much higher resolution clinical disease trajectories that will give even more information on the phenotype of each of these donors. And also, yeah, as I mentioned before, we want to work on digital pathology because I think that's ultimately where we need to go. Try to get the human out of the equation as much as possible. Yeah, I acknowledge the people in my group, Nienke Mekkes, Erik Hoekstra, Alize de Boer, Ekaterina, Shivan Kumar is going to start next month. And also my collaborators at the side of the Netherlands Brain Bank, Inge Huiting, as well as the neuropathologist Sanne Vermorgen, Annemieke Rozenmuller, and Nina Fransen. Annemieke just retired a few weeks ago, so we're very lucky that we have two neuropathologists taking over. They're a rare breed and they're becoming more and more rare. So if you're a neuropathologist, you are pushing the boundaries of science forward, and please don't change career because we really need you right now. And yeah, I think that's what I wanted to share. Oh yeah, last thing, obviously, I should always mention many funding sources in our way that have been able to support these and other activities. So it's really a pleasure to be back here in San Diego and to talk about my work. Thank you for your attention.